study, DNA methylation of Criostridia gigas in response to ocean acidification. And so I know that you guys are already all asking yourself, why do we care about Pacific oysters? Let's see here. I'm gonna go out of order. So why do we care about ocean acidification first? So basically from 1751 to 1994, the ocean pH has dropped from 8.25 to 8.14. And since pH is on a logarithmic scale, this represents a 30% increase in hydrogen ions. And the issue with this is that it's occurring 100 times faster than it has in the past 20 million years. So this is happening really quickly. Basically, specimens, or I should say different species, aren't going to have time to adapt. And there's not really anything we can do about it. We either need to find ways to sequester carbon or ways to reduce our emissions, and this probably isn't going to happen. So we might as well study it, and we might as well figure out what the biological implications are of ocean acidification. And one important thing to note is that invertebrates are what are really going to be affected by this ocean acidification, such as the Pacific oyster. So this map shows everywhere where Pacific oysters are cultured, so it's all over the world. It is the most widely cultured organism in the world at 4.38 million tons. And basically, so one of the reasons is people like to eat it, and it's really gained popularity in the past 20 years. And additionally, it's been put in place to displace some natives that might not have been doing as well. And so in addition to its economic value, it also has this huge ecological value because it creates an environment for other organisms to live in. And then another layer to all of this is epigenetics. Basically, this term was coined in 1942, but research into the area hasn't really started until recently, like the past 10 years here. And we don't really understand, completely understand the role of epigenetics in invertebrates. So doing so would allow us to draw conclusions to vertebrates. And also there's implications for different diseases that are related to epigenetics in humans, such as Angelman syndrome. So looking at invertebrates and looking at epigenetics, we can start to kind of draw larger conclusions with implications for human biology.
So our objectives was to quantify the physiological response of diploid versus triploid oysters when exposed to different acidified conditions. And as you heard previously, we won't talk about the treatment because they're the same as the gene expression, protein expression group. And we also wanted to discover the implications for varying levels of DNA methylation, whether that was related to coping with acidic conditions. So, our methods, like the other group, we used dot plots, and Andy used methylated cytosine dot plot, gel electrophoresis, restriction enzyme digest, PCR, and qPCR. So restriction enzyme digestion, if you've forgotten, is using bacterial enzymes to cut specific recognition sequences. And in this case, we use restriction enzymes that can cut or can't cut at methylated sites. And MSP1 was the one that can cut methylated sites, and HPA2 was the one that couldn't. And the methylated cytosine dot plot is confirming the presence of a methylated cytosine with antibodies or probe binding. And gel electrophoresis, if you've forgotten, is separation of DNA fragments by size using an electric field. And it's...
Okay, so we're going to go to our results, and we're starting with our mortality table with our treatment. And as you can see, we saw no mortality in our control group. However, on day 1, we saw that 5 diploid and 2 triploids had died, so immediate tissue sampling had to be taken place. restriction enzymes. This is for Jagesin 2. The top columns are diploids and the bottom columns are triploid. And the right-hand side columns are MSP1 and the middle is control. And our left-hand side columns are HPA2. We also looked at HSP70, and for this one, the top columns are triploids and the bottom columns are diploids, and the right-hand side column are HPA1, and our middle columns are MSP1, and the left-hand side columns are the controls. And what was different in this gel was that there were smaller, fainter bands in all our MSP1 samples. And we also did quantitative PCR for the Jagesin 2 gene. And we couldn't do qPCR for our HPA70 because our primers were not the right product size. So in our control samples for Jagesin 2, there was an expression level of 1.3 to -7. HPA2 had 2.5 to -8, and our MSP1 samples had 1.5 to -8. With our methylated cytosine dot plot, each sample had 400 nanograms of DNA, and we saw that most of the individuals in the low pH conditions had higher levels of methylation, and also all of the diploid oysters showed levels of methylation.
Alright, so some conclusions. What can we start to conclude from these results? So just first thing of what went wrong with our experiments, I think we said that our blooper reel would probably be longer than like our actual film if we were to look at all this. But that's okay, because we still definitely saw that there was this difference in methylation in both the restriction enzyme digest and in the cytosine dot plot. So, like I said earlier, it's not really like mechanisms for this sort of stuff isn't super well understood in invertebrates. We can hypothesize this has to do with phenotypic plasticity, so it's modifying the DNA, it's methylating it, and it's probably going to express genes in a different way. And obviously this would be a response to a stress such as acidified conditions. Additionally, we might assume that they're going to be able to pass this stuff on to future generations, since that's what occurs in vertebrates and other examples of things that methylate. And basically, I should say for diploids and triploids being the same, that has implications for the aquaculture industry. So basically they can kind of rest easy. There isn't this differential between diploids and triploids. So they're going to be just fine farming triploids as they would be having the natural diploids. And looking at next steps, it would be important to do a more like long-term chronic exposure kind of study, just because this would allow us to get basically be a more natural way of looking at the system than kind of just doing this short-term high exposure. Additionally, we want to couple DNA methylation with looking at things like protein expression and RNA expression, kind of like the other group did. So we probably kind of want to look between the two and make comparisons about how methylation is actually affecting the gene expression in these oysters. And it would be nice to look at other organisms closely related to oysters, maybe clams would be a good example, and kind of start drawing comparisons, which would kind of just allow us to gain a better understanding of methylation as a whole and how invertebrates use it. And I think that's it. So thank you.